Welcome everyone to this week's edition of the EV Journal Club. As you can see from my background, I still have my ICEV 2020 background, even though ICEV 2020 Live was over last week. And that's because the content is still available for 2 more months. So if you have not registered for ICEV 2020, you can still do it. And if you have plenty of time to look at the posters and oral presentations and also the recordings of the live sessions. So thanks everyone for joining, and I'd like to send a special thanks to our presenter today, who is Catherine Millar-Haskell from the University of Delaware. She's in the Gleghorn lab there. And so Catherine, thanks so much for joining us. We're looking forward to your presentation. And we also have several of the authors of the preprint on the line today. So as I understand, we have Andre, Martin, Heidi, and Fausto on the line. And I'm going to unmute you guys and just ask you to perhaps tell us a little bit about this paper and how it came to be. I understand that this is the first preprint from your group. So first of all, how was the process of the preprint? Why did you decide to make this paper a preprint, and are you happy with the outcome?
Hello, Ken. Can you hear us?
We can hear you loud and clear.
Perfect. Thanks a lot that you have chosen our preprint for a journal club. It's a great value for us to get input and criticism to our paper. We actually started in the vesicle field like 3 years ago, because in this project we've had the hypothesis that extracellular vesicles could be an important mediator of the effect of a clinical product that we want to investigate. And that's what the paper is about. And we actually submitted this paper to the Journal of Extracellular Vesicles and got rejected and got some
hey, it's happened to me too, don't worry.
From the reviewers that we should further improve the manuscript, and as we wanted to have it out, we deposited the manuscript as it was on bioarchives, and meanwhile did some more experiments, did an animal model and further investigations about the mechanism. And we actually plan to submit the manuscript to another journal within the next few months. And so very happy about further critical input to what we could improve to get this story more clear and more understandable.
Excellent. Well, I'm really glad that you and the team could join today, and I'm actually going to make you a co-host so that if there are any questions that come up during the call, you can answer them. Catherine can answer them. You know, whoever's best suited to give the answers. So thanks again, and so Catherine, I will hand it over to you, and you can go ahead and start sharing your screen.
Perfect, thank you. So everyone can see it and hear me, okay?
Looks good.
Perfect. All right, so I will be presenting this paper entitled Extracellular Vesicles from Therapeutic Grade Allogeneic Human Placental Stromal Cells Induced Angiogenesis and Modulate Immunity. So a little bit of background on the disease that is being studied. Peripheral artery disease is a fairly serious disease that involves blockage of arteries, most notably to the limbs, but most significantly to the legs. This, if left untreated, and if it worsens, it can lead to what is called critical limb ischemia. And when this develops, there's a high rate of amputation and risks to both cardiovascular health and the immune system. Some of the comorbidities include a high risk to infection, transient ischemic attack, and things involving poor blood flow to the organs. So with this in mind, the focus of the paper is to look at a product The product itself is actually a cell line called PLX, or placenta expanded stromal cells. These are mesenchymal-like stem cells that have been investigated for their ability to initiate angiogenesis and muscle regeneration in critical limb ischemia. Right now, they are currently still enrolling in the phase 3 clinical trials. The cells are owned by a company called Thurstem, and this is part of a greater initiative called PACE. PACE is a project in which the goal is to evaluate the efficacy of these PLX cells and to promote additional studies to kind of investigate the mechanisms behind why these cells can improve the outcomes of patients with critical limb ischemia. So just a little broad overview of the paper. The idea here is that the PLX cells are injected intramuscularly into a leg, and the cells are thought to secrete a number of soluble factors that lead to pro-angiogenic potential and immunomodulatory behaviors. And so here, the authors propose that extracellular vesicles might be involved in some of this. So the goals of the paper are to essentially formulate a method for separating out the EVs from soluble factors to reduce the contamination from the EV fraction, and then to robustly characterize these samples. Then part of the goal is to kind of identify some of the proteins that are unique to EV fraction and to the soluble fraction to try to tease out which proteins involved in immune function and angiogenesis might be involved. And then finally, to measure some of the functional outputs of the EVs and the soluble fraction. The first thing that was done was a robust characterization of the media. So here you can kind of see a panel where 3 different media formulations were used, basal media, the alpha MEM, EMEM, and also EGM2. And what the authors did was they chose 2 different serum supplements, FBS and HPL, or human platelet lysate, and measured the concentration of particles in the media. They went and then further initiated a clotting of the media by adding fibrinogen and then removing that clot, which is denoted by this asterisk. And then they further attempted to deplete the particles from the media by using 2 techniques, UCF and TFF. So just showing here that there is a 2 orders of magnitude decrease of particle concentration after the particle depletion technique. And they also looked at some serum-free defined media, some chemically defined supplements, and went ahead and did some more characterization by culturing the PLX cells in the media for about 6 days, and then looking at the change of particle concentration. So as you can see here, with the particle depleted media, there is a marked increase in the number of particles that are produced after culturing with the cells. And you can see the same changes occurring in this particular supplement called Nutristem. And then finally, there's just a time course experiment showing the increase in particle concentration from day 0 all the way to day 6. And so I just want to point out here that the 2 things that are in consideration for at this point are the alpha MEM that's been depleted by TFF, and then also the Nutristem. But then upon further characterization, the authors decided to choose the alpha MEM depleted media because the Nutristem seemed to have a more heterogeneous distribution of particles, and the particle size was also significantly larger. So for that reason, for efficacy and efficiency, the authors went with this media. So just for people unfamiliar with TFF, I just want to give a brief overview. It's tangential flow filtration, which is a type of ultrafiltration in which you add media to an inlet and then induce a cross flow through a filter and then an outlet. So what happens is for whatever molecular weight cutoff you have for the filter, the smaller particles will pass through, and everything that passes through that's smaller is called the permeate, while everything that is retained is called the retentate. So this is kind of important for this specific experimental flow through, because what authors did was they used the alpha MEM media and then did the particle depletion prior to culturing the cells for 48 hours. So then after 48 hours, the conditioned media was collected and it was passed through TFF with a 300 kilodalton molecular weight cutoff. And the soluble fraction, or in this case, the permeate, was kept, while the crude EV fraction was passed through 2 more times in wash steps to purify it further and to remove any extra contaminants, lipids, and proteins. So just to summarize, there are 3 fractions that are being studied throughout this paper: the soluble fraction, the crude EV fraction, and the purified EV fraction. So here's just a transmission electron microscopy showing the morphology of the EVs. As you can see, the crude EVs and purified EVs are both look good to me. And on the left, you can see the PLX-derived EVs with a smaller magnification. So next, authors did Western blot. The first thing they wanted to look at was whether they could determine lineage specificity from the EVs. So they took 3 different PLX donors, those EVs, and compared it with endothelial cells, and looked at 2 different markers, CD81 and CD9, which are both EV markers, I believe, and found that there was more CD9 in the endothelial cells and more CD81 in the PLX cells, the EVs. And so for every Western blot, they did a corresponding quantification of the luminescence, which is the thing you can see on the right. So here I broke it down to the EV markers. So they did 4 EV markers: the CD9, CD63, 81, and flotilin 1. And as you can see, the crude EVs and purified EVs both contain the tetraspanins, but the purified EVs have more of those markers and also contain flotilin 1, while the crude EVs have a very faint signal, if anything. They also did what you might call negative markers of EVs. So they chose calnexin and GRP94, which are the endoplasmic reticulum and the Golgi, respectively. So the cell fraction should have a very dark band, while the EV fraction should not, and that's what they saw. Additionally, they looked at ApoA1 and human serum albumin. The human serum albumin is what you would find in the soluble fraction. So the point here is that there's a lot of it in the crude EVs, but they were able to get I think up to 70-fold reduction of HAS in the purified EVs, demonstrating that they're able to remove the majority of the protein contaminants from their pure EV fraction. And then here is just an extra stain showing that this band that they saw repeatedly in each of the fractions turned out to be CD81, and they just quantified the relative purity and found that as they went towards pure EVs that there's an increase in this. So finally, we get to a little bit more of the protein analysis, the proteomics. This was a bead-based multiplex assay using flow cytometry, and they chose a panel of different antigens to investigate the relative abundance on these EVs. And I guess just to point out, they confirmed that CD81 and CD63 are highly expressed on the purified and the crude EVs. And there's some other ones that are kind of moderately abundant, including a few fibronectin receptors, a few ECM interaction proteins, one tissue factor, and one cytokine. So to kind of investigate the single EV, to measure single EVs in flow cytometry, they did a size calibration and fluorescent signal calibration. For the size calibration, they took silica beads of different sizes and then used that to gate the EVs. So when they flowed the EVs through, they were able to get a size gating. And additionally, they did the same thing with two fluorescent stains, the calcine and lactodherin. So just to go a little bit more into this, essentially all of the EVs that were that had a positive fluorescent signal, 98% or more of those EVs were positive for calcine, which presumably means that the EVs came from intact live cells. And the ones that stayed positive for lactodherin presumably came from apoptotic bodies or cells that were not intact or dead. And then shifting a little bit more into protein analysis. This is just a schematic showing the overlap of proteins that were found in the different fractions. As you can just see here, the pure EV fraction has 258 unique proteins, while there is significant overlap, but there's also a lot of non-overlap. So to look into that a little bit further, there's a pre-analysis that was performed using kind of a modified dot blot array using Western blot. And here, the two things that were compared was just the medium compared to the soluble fraction and the crude EVs compared to the purified EVs. I don't want to get too much into the specific proteins, but just to note that there are quite a few pro-angiogenic proteins that were present in the purified EVs and quite a few that were also present in the crude EVs. And just to note that the soluble fraction had fewer relative abundance of some of these proteins. So here, this was a quantitative characterization by using tandem mass tag. Over 1,000 proteins were identified and collected here in this heat map. I want to point out that you can just kind of see the relative fold changes in each of the donors and each of the fractions, while with the proteins that are in low abundance, the UVs are highly abundant in the soluble fraction and vice versa. There's also a volcano plot, which is basically just looking at statistical significance compared to fold change. And the blue markers up here and around indicate proteins related to the immune system, while the red is angiogenesis. So it's just another way of visualizing it. And then finally, here's just a table showing the various proteins that are present in the purified EV fraction. There are quite a few that are related to immune modulation, quite a few also for angiogenesis, and as you would expect, a lot of extracellular vesicle markers that are present. So I just want to get into some of the functional assays. The first thing that was done was the angiogenesis, which is just using matrigel and seeding cells, endothelial cells on top. And on the left, you can kind of see various different concentrations of the fractions that were dosed in culture compared to the endothelial cell. So for example, 10,000 particles to one cell is the highest amount. And you can see that all three fractions have some effect on vascular network formation, and that this seems to be dose dependent. And on the right, you can just kind of see the images of what a robust network looks like and what no network formation looks like for the lower concentrations. Next, they did a couple assays to look at immune function. So the one on the left is a PHA-induced T-cell proliferation assay. So they added in about 5 micrograms per mil of PHA, which is a chemical inducer of T-cell proliferation. And just like with the angiogenesis assay, this appears to be dose dependent, and the crude EVs and pure EVs, along with the cells, are able to inhibit T-cell proliferation at a high concentration, which seems to be reduced with lower concentrations. And what's interesting, and I don't fully understand this, the soluble fraction doesn't appear to have any effect at inhibiting T-cell proliferation. So maybe the authors can talk about that a little bit later. On the right, this is just showing a different immune assay called the mixed leukocyte reaction, or MLR. This is done by mixing leukocytes from 2 different sources, two different hosts, and then mixing them and then investigating the reaction and subsequent inhibition. So as you can see here, the pure EVs appear to not only not inhibit T-cell proliferation, but maybe perhaps boosted it a little bit, which was interesting. While the PLX cells at high concentrations were able to get a near 100% inhibition. And finally, there's a little bit of thromboelastometry that was performed. And I just want to go over this figure just a little bit more carefully, just in case people are not familiar with thromboelastometry. So there are a couple definitions out here. There's the clotting time, or the latency time between adding the reagent to start clotting, and then the formation of the clot, or the start of the clot. Then there's clot formation time, which is the time from when the clotting time starts to when the clot reaches 20 millimeters of firmness. And then there's the alpha angle. Now this angle is just the tangent between the 0 millimeter, the start time, and then when the clot reaches 20 millimeters. And then there's the maximum clot firmness, which is just the greatest, the maximum vertical amplitude. So as you can see here, the maximum amplitude is roughly 40 millimeters. The clot formation time is denoted by this hot pink shaded area. And the alpha angle can just be drawn from 0 to 20 and measure the angle of that. So here's just a quantitation of those measures. As you can see here, the alpha angle is fairly high for the cells, the crude EVs, and the purified EVs. And between them, there's no significance. But there is significance with the clotting time and also the clot firmness. As you can see here with the clotting time, the crude EVs and the purified EVs took a little bit longer to initiate clotting compared to the cells. And with the clot firmness, the crude EVs and the purified EVs were not different from each other, but were significantly lower than the cells. So with that in mind, I just want to summarize this paper briefly. This preprint basically outlined an efficient way to isolate and to produce and isolate EVs, suitable for doing multiple experiments and for doing proteomic and functional assays. So interestingly enough, the TFF depleted media was more effective at producing EVs and reducing particle contamination for downstream analysis while maintaining cell culture. The proteins that were found in the soluble factor were significantly different than the ones found in the EV fractions. The crude and the purified EV fractions and also the cells were able to induce vascular network formation, and that these were done in a dose-dependent manner. And finally, with the immune cell assays, the EVs were able to inhibit T-cell proliferation with the induction experiment, but that the purified EVs were significantly less efficient at it, and that the purified EVs didn't appear to inhibit T-cell proliferation in the mixed leukocyte reaction assay. And that's all I have.
All right, wonderful. Thank you so much, Catherine. We will move to some questions now that have come in through the chat box. So I guess, let's see, how should we do this? I'm gonna, first of all, let people unmute themselves so that they can ask their questions. And we'll start, I know that there has been a little bit of a conversation already in the chat box, but why don't we just bring that all out and let people have a back and forth here as well. So Lilia, you asked the first question. Do you want to tell us what you were thinking about with EM versus other quantitation or sizing techniques?
Yes, thank you for the presentation. I think it's really interesting, and especially the TFF usage. I have just a very technical question. What was the reason of using TFF 300 kilodalton? The reason why I'm asking, because we've been working with 300 kilodalton pass membranes to concentrate the cell culture supernatant by the Sterilitech device, as many of us, I guess, been doing. And then we've seen the publications, several publications from Ann Kendrick's group, and also we tested in our lab that we actually lose some vesicles, a lot of vesicles by this type of the concentration. And I'm just wondering, what about the TFF in that case? Because we decided just with the same strategy to go for purification of the soluble fractions using the two devices, TFF 200 nanometer and TFF easy from Hansa, which is like 20 nanometer cutoff, just to be completely sure that we got no vesicles in retentate or in, not actually in pyramid. So, and that's why I asked about the electronic microscopy pictures of the soluble fraction sample and TRPS with NP 800 and whatever NCA. So just if there is any explanation for 300 kilodalton so far.
Can the others answer that?
Maybe I can answer the question, because we were using the 300 kilodalton because the pore size according to the manufacturer. So we are using Repligen TFF system. They say that the 300 kilodalton membrane is about 50 nanometers in size. So we wanted with this size to really keep more or less all the EVs inside the column, while still be able to efficiently remove soluble proteins in the washing step. Because when we were using smaller columns, we saw that proteins and also small protein aggregates are also retained in the column, and we cannot wash them through the smaller filters. And we were using the TFF system because it's much more efficient and scalable than, for example, ultracentrifugation. As someone asked in the chat box, we start usually with 0.5 liter to 1 liter of culture supernatant to produce our vesicles from, and therefore also have really high concentration factors between 100 and 200 fold concentration to give us sufficient vesicle numbers and concentrations that we can use then for the functional assays, because you need really high concentrations to get good results then.
Okay, so you actually haven't measured the particle count and the size distribution of whatever it is in the soluble fraction sample, right?
We measured the particle concentrations also in the soluble fraction, so the flow through of the membrane, and cannot detect particles there. They are below the detection limit. So we cannot say anything about size, but with the TRPS that we are using, we more or less are at the background level of the buffers. So we cannot see any particles there. So the system should be tight and not leaking vesicles.
Okay, thank you.
Great, okay, thanks for that. Phil Askenaz, a question about the bead-based assays.
Yeah, can you hear me?
Yes, we hear you, Phil, thank you.
So I have a few questions. My first one was, how do they know there was one EV per bead when they did the flow cytometry?
The bead assay that we performed is not single EV based, it's a bulk based assay, and it was described in a publication by Mark.
So you can know you're looking at one bead, but you don't know how many EVs are on the bead.
You don't. You're just looking at the fluorescent intensity.
Yeah, that's the problem with beads.
Well,
so
it's a problem if you want to do single EV analysis. On the other hand, this is a way to understand the overall levels of a particular antigen.
Oh, definitely, definitely. Yeah, that's what we do. So what's the throughput? How much time does it all take? And what's the loss? So compared to what you start with, what percentage do you end up with, and how long does it take to do the whole thing?
You mean the purification process?
Yes.
Well, the most time-consuming step is the cell culture. We use cell factories in big scale. This takes about 1 week or so to get sufficient amounts of supernatants. And the TFF process is done in, I would say, half a day.
And the loss?
Efficiency, we, with the TFF, can about get, I would say, roughly 90% of the vesicles that we have in this conditioned media, we can retain within the TFF system.
That's very good. Thank you.
Thanks, Phil. Was that all of your questions?
Yeah.
Okay, perfect. All right, then I think we have some other questions about some technical aspects here. Anoop, are you on the line? Do you want to ask your question about the volumes and reductions?
Hey, thanks, Ken. Yeah, I think can you hear me?
We can hear you. Thank you.
Oh, right, yes, yeah. So like pretty much it has been answered. So the starting volume is like 1 liter, and then the final output would be around 10 ml. Is that the final output for these EVs?
Yes, that's correct.
Right. Okay. Perfect. Thanks.
So maybe related to that, like what? Juan, Juan, here, are you on the line? Because I think you might have an extension to that question.
Yes, I am here. Yes, the question is how long take to have the media with this TFF system?
As I said before, we culture the cells for about 1 week and harvest every second day. And then the TFF process, we have for the 1 liter about flow through the membrane of about 30 to 50 milliliters per minute. And therefore the concentration step takes about 2 to 3 hours, and the washing step afterwards again, I would say another 2 to 3 hours. So the whole concentration and washing process is done in about half a day.
Okay.
So it's a quite efficient and straightforward and also very nicely scalable method compared to ultracentrifugation, where you are very limited with the rotor sizes and also have really long run times.
Mm-hmm. Yeah. But had you noted any different, let's say, enrichment in tetraspannings? For example, I saw some image, yes, that for example, regarding the ultracentrifugation and the TFF, that you lose some subpopulation of vesicles, or you are enriching in one and in another. So had you noted anything about that?
We haven't had a look at subpopulations of vesicles, unfortunately, and also in this project didn't directly compare different purification methods. But a colleague of mine with another project is at the moment comparing different purification protocols and could not really see different enrichments of different tetraspanning markers with the different purification methods. It's, from our point of view, rather the cell source that makes the difference than the purification method per se.
Okay, thank you.
So maybe we can even talk a little bit more about the actual setup for the TFF. So the question came from Olesia, which TFF device did you use? And then I would add to that, what did you look at other ones? You know, what was it that made you choose what you used?
We are actually using a key-eye system from Repligen. So it's a standalone peristaltic pump with a pressure sensor, a restriction valve, and in this case, hollow fiber filters, which we actually preferred over the traditional cassette filters because you have better cleaning capacities for these hollow fiber filters. You have a better flow characteristics of the hollow fiber filters, and also you have less dead volume, which is very important if you want to concentrate the vesicles to very low volumes to get high concentrations. We also had a look at similar systems from other suppliers, but due to the easy use of this system and the quite low cost of the spare parts and consumables, we have chosen this system, and it's also quite convenient to use in our hands.
Yeah, that's good to hear. And I would just note that TFF, there are a wide range of options that are available, everywhere from just using your own peristaltic pump with a disposable filter set to you know, these standalone things that also have downstream sensors that will let you monitor what's coming off at any given time. So that it is indeed, you know, there's probably a solution that would work for most labs depending on how much money they're able to put in. So that's good to hear. Let's see, we have, oh, and this is actually a very interesting question. I'm kind of going out of order here, but Matthew Hudson asks, why harvest EVs every 2 days versus every day? Of course, this is not about the technique so much as the question about whether EV release changes depending on the concentration in the media and so on. So have you looked at different harvest times and whether that affects either the yield or the function of EVs?
We have looked at different harvesting time points and actually found that cells are producing EVs, let's say, in a quite linear range the first 2 days. And if you culture the cells for a longer time in the same media, they start, especially at high densities, they start starving and then start to use up their own EVs as a kind of nutrition maybe. And so after 2 days, the production or the increase in EV amount starts to reduce. And why we choose 2 days and not 1 day is because cells also need like growth factors and stuff they produce on their own. So if you change the media too often, the cells are also not too happily growing. And due to the fact that we couldn't see any or too much decrease in EV numbers within the first 2 days, we decided to harvest every 2 days the media. And actually we are not harvesting all the media. We leave half of the media still there so that they are not completely in fresh media all the time, that they still have some of their, let's say, goodies and known environment around that they need for best growth conditions. Because we think when we keep cells happy during EV production, compared to many other people that just throw the cells in complete serum-free media, where the cells more are unhappy and start maybe dying or going to apoptosis and more produce apoptotic bodies. In our hands, we see that when we grow the cells in good conditions, we hope to really get EVs that are produced by functional cells that might then have other effects than if you study EVs from cells that are not that happy.
So what are you monitoring the cells then constantly or regularly? And this relates to a question from Camille who asks about confluency. So are you measuring confluency? Are you measuring other outcomes?
We have a look at confluency of the cells. So we first expand the cells like for 1 week. And then when they're about, let's say 60–70% confluency, we start adding the vesicle-free media or vesicle-depleted media, and then start their harvesting process. And then in the end, the cells are like 100% confluent when we finally harvest the cells, because the cells are adherent, so we can just change the media during the days before. And then when we do the final harvest, we harvest the cells and have a look at that cell percentages, and these are usually below 5%.
Good, all right. Another question from Mandy Chan. Mandy, do you want to ask this question? You have a few questions in here, actually.
Sure, thank you. I have some experience with TFF, but not in the context of extracellular vesicles. I'm curious, why do you choose 3 different TFF steps instead of one prolonged one with washing, assuming the purity would increase with higher wash volume? And do you switch to a new TFF cartridge every step? Is it because you have some clogging issues? Yeah, I just want to know your experience. Like, are there challenges as you optimize your workflow?
I actually do not 100% understand what you mean with 3 steps, because for us, it's like we put the whole conditioned media into the TFF system in the reservoir, and then make a constant feeding procedure to the smaller reservoir, where we first concentrate the vesicles, and we then after having concentrated the vesicles in the same column, do the isovolumetric washing process. And we do not change the filters in between. We actually also reuse the filters. We have a quite rough and consistent washing protocol involving sodium hydroxide solutions and ethanol to keep the columns sterile, and therefore we can reuse them several times. We see actually when during the concentration process, we see that we get some kind of fouling of the membrane, because the flow through is reducing when we get higher concentrations, but we do not really clog the membrane during the process. So it's the flow is reducing, but otherwise it's working fine. And as we can wash the membrane and reuse it, this is not a big issue in our hands.
Are you pre-blocking?
No.
No pre-blocking of the membrane. Okay. All right. Okay, so a few more technical questions have come in before we get to some questions about the assay itself and then storage. So EV-free medium that you're using, is that something that you prepare every day, or is it something that you can make and then store? That's a question from John B.
Actually, the EV-free media, we prepare in big batches, also with the TFF system, and then just freeze it down at -80 actually, and have it off the shelf.
So you run the media through there, not the individual components.
No, we actually prepare the media completely ready and then run the whole media through the system. It's actually necessary because if we would run the individual components, the biggest issue is the platelet lysate that we are using, or others might use FBS, because also for people using ultracentrifugation to deplete vesicles, you have to dilute the platelet lysate or FBS to a certain amount, because otherwise the consistency is blocking either the membrane or causing lipid phase separation in ultracentrifugation. So in our hands, it's most efficient to really prepare the complete media and then filter the ready-to-use media before user actually make it in bigger batches and store it.
Right. Good. All right. Well, thanks for those answers. Catherine, did you have any comments you wanted to make on technical aspects of TFF, or any questions that you might have?
I had questions, but I don't remember right now.
Okay. All right. Well, yeah, let's. I think I'll ask Nikki Norenhouten. You had a question about the T cell proliferation assays.
Yeah, I just had a question about whether for the T cell proliferation assays, was it a single dose of EVs or was it multiple doses? Like, because I think it was a 4-day assay. So was it given on day 1 and then you assayed 4 days later?
It actually was only 1 dose that we directly added the EVs at day 0 when we prepared the assay. But we also tested for a longer assay period where we were using not a PHA as a mitogenic stimulus, but rather the mixed leukocyte reaction as a kind of graft versus host mimic situation, where we added the EVs every day, but couldn't see any difference in the outcome of the assay.
Okay, thank you very much. Great presentation.
For the PHA, the 1 dose is sufficient.
Cool.
So I think a question was then also a question about passage number of cells. So how long can you keep these placental cells alive? And do you say, well, after this point, we don't use the EVs anymore? Do you notice differences?
We are actually taking the placenta cells that are highly expanded from the company Pluristem, and in most cases only use the clinical grade cells and expand them once. So we do not make our own stocks, and therefore, because I do not exactly know the number of passages that these cells have for the production step, so I cannot really answer how long you can keep these cells in culture, but due to the fact that Pluristem has a patented process where they use one placenta to make 300,000 doses of 100 million cells each dose, that these cells are really highly expanded in multiple steps involving multi-layer flasks and also bioreactor expansions.
Great. Okay.
Sorry, can I add to that just real
Yes,
quick?
please.
Does this mean that you are not given a passage number? You don't know how many passages have occurred?
Exactly. We do not really give them a passage number because we usually saw the clinical grade aliquots, expand them once, produce vesicles, and after the vesicle production, we harvest the cells, analyze the cells, and discard the cells.
Okay, thanks.
So we're coming towards the end of our time here today, but we do have an interesting question from Steve Ellman, who asks, how are purified or even crude EVs stored and buffered prior to functional assays? So this is actually a very big question, and I think a lot of people would like to know if there's a single answer to it, and unfortunately there probably is not. So maybe this is an opportunity for us to have a broader discussion here, so we can hear from the authors. Maybe we can ask Catherine what she does. And we can even ask anybody on the call if they have particular storage methods that they prefer. So let's start with Salzburg. What do you guys do?
So we store the EVs in 0.9% sodium chloride, so physiological saline solution with 10 millimolar HAPES as a buffer added. And we snap freeze the vesicles in small aliquots and store them at -80 degrees. That's a protocol that we got recommended by Mario Cimona and also Bernd Giebel. And in our hands, this protocol is working. And to be honest, besides comparing the vesicle counts of fresh and frozen EVs, we have not further investigated different storage conditions so far. But maybe some others have other ideas or even suggestions what works better or how they do.
I know there are different buffers you can use. Some groups use DMSO, about 10%. I usually use my EVs fresh, and if I have to freeze them, I limit the type of assays I use for those EVs, because I assume that a majority of them will be lysed or not intact, which is important for my experiments.
So I've put up a really quick poll here. So people on the call, let us know how you store your EVs. It's kind of silly because I didn't have time to really think about it very much. But I guess you can see it on the screen. But do you store your EVs refrigerated at 20 C, at -20 C, -80 C? And do you store them in a cryoprotectant, physiologic salt, or water? So if people want to go ahead and say how they prefer to store, that'd be great. And in the meantime, while answers are coming in, does anybody else have some comments about how they think EVs should be stored? Everybody's shy today. I know this is a question that a lot of people have feelings.
Can I say something?
Absolutely.
Yeah, thanks. I'm just wondering about the large vesicles and small vesicles as usually. So I know that for the small vesicles, it's not even the problem if you lyophilize them and kind of not for a long time and then try to make some assays. But for the large one, it's still a challenge to me to get the really good the particle number and then functionally analyze the large vesicles after this like standard procedure when you freeze it at -18 PBS on HIPPS. So when I was just wondering whether somebody tried out the method pretty close to the cell storage instead, because the large vesicles are pretty big, and I think osmolarity doesn't really help to keep this large membrane still stable by freezing and thawing. And I'm just wondering if somebody tried to store the large vesicles like cells, so with some DMSO maybe. No idea.
Yeah, well, I think that makes, it makes sense that the larger the vesicle, the more it's going to be behaving like a cell under, in terms of how we would want to store it. And I agree with you. It seems that smaller vesicles are more stable, I guess, until you get down to a certain membrane curvature at the very smallest end. And they seem to, I mean, we can even store them in water if we want to, most of them. But I don't know if that's advisable. It looks like so far we have 84% of our respondents who are saying that they store their EVs in physiologic salt, in some kind of a salt buffer. Whereas 7% are using water and 9% are using a cryoprotectant. Of course, there are a lot of cryoprotectants that could be used. DMSO, Trehalose is one that came up in the chat box and that has been published on. Some sugars, and then lyophilization. So Lilia mentioned lyophilization, and Genevieve says that she was advised to lyophilize and saw a review about lyophilization in Trehalose, but has not tried this yet. Let's see where, yeah. But the consensus seems also that everybody wants to store these, not everybody, but most people want to store them at minus 80. Unless you're using them right away. Any other comments about storage? I don't think we have a consensus in the field on this.
The only thing also...
Oh, sorry. No, go ahead.
To have in mind that what is going to be used these exosomes or these vesicles. Because, for example, for metabolomics, you cannot do resistant buffer with high salts, because this will be useful for the machines. So this is also to have in mind, which is the downstream useful.
That's a very good point. Very good point. You have to keep in mind what you're going to do with them later too. Yeah.
Yeah, I was going to add to that real quick along the same lines. When we're doing this for like surface analysis of transmembrane proteins in the vesicles, we store them at minus 20 and in just 1x PBS, and that seems to maintain the vesicle intactness enough for studying those. But if you're concerned about contents, then you may have to store them differently.
Thanks, Killian. And anybody else want to comment on this?
I can add a little bit. My name is Alessia. I'm from UNC Chapel Hill. Actually, we store our vesicles in PBS, and I have 1 vial of vesicles which were isolated from January, and then still stable in size and concentration from like 6-7 months already. So I didn't check the protein expression yet, but I'm going to, and it seems they're very stable in FOSI and PBS. And we also did some study on presence of DMSO. More than 5% DMSO will totally lead to vesicle segregation. So just clumps and like precipitate during time. So I don't recommend DMSO like in hormones. But if you want to preserve your exosomes, we usually store in minus 80 and they stable in size, by size and charge and concentration for more than 6 months. We tested them. We even injected these exosomes, and they like very similar to freshly isolated exosomes.
Very good. So we're at the end of our hour now. Are there any final burning comments? I know we have a few questions that are in the chat box that we weren't able to get to that are a little more complex. But Catherine, do you want to make any closing comments?
I guess just to say that I just found the paper really interesting, and that I hope that TFF might be a good way to maybe further explore how to separate the fractions and purify them.
Very good. All right. Well, Catherine, thank you so much. We really appreciate your contribution today for taking us through this paper. And also, I'd like to thank the authors for joining us and for sharing their insights into their work. Who can do it better than they can? So thanks, everyone. I hope everyone has a great rest of the week. And we will look forward to seeing you again soon.